I hope everyone's having a good day so far.
Can you use the microphone, please?
Yeah, I'll grab my hand up one. Is that any better? Excellent. Okay, so a few introductions. On stage I'm Russell, this is Pasco and Joy, and obviously in the bright green t-shirts, the rest of the team. We're comprised of 3 biomedics, 3 molecular biology and geneticists, and 1 biologist. And of course, the man that's organised it all, Richard Kelwick, is our PhD advisor. We're in week 6 out of our 10-week project. Everything's going quite well at the moment. I think we're enjoying it, and we're getting there. Okay, so the presentation is going to take sort of a timeline form, going from the sort of the broad range of ideas from the top all the way through to where we've got to now with our project. So our university looks at has a lot of research groups that look at nitrogenous species and nitric oxide sensing and that sort of thing. They have a lot of problems because a lot of these species are quite unspecific, or non-specific rather, and quite difficult to sense. So we thought we could try and use synthetic biology and iGEM as a way to try and overcome these problems. The main molecule we're looking at is nitric oxide. It's an extremely reactive molecule with about 20 to 30 second half-life, and it just tends to react with everything it can do, which makes it really difficult to sense. Adding to that the problem of most promoters and mechanical ways of trying to sense it also sense anything else with nitrogen and oxygen in it, for instance, nitrites and nitrates. So in order to get a good view of the nitric oxide in the system, it's particularly difficult. We've looked at the application of nitric oxide, and it has a lot of great applications that can be useful for physiology or for the environment. But obviously, if you can't sense it accurately, it's difficult to be able to tell what's going on. So we decided to uptake that challenge and see what we can do with it. The first promoter we looked at was called PYEAR. It's a promoter found in E. coli in Lactoperon, I'm not going to try and pronounce it. And it's one of the more specific ones for nitric oxide. Unfortunately, in the great scheme of things, it's still not particularly specific. It's mainly activated by nitric oxide, nitrites, and nitrates, whereas we just want to look at nitric oxide itself. We decided to look at it anyway to see if we can get out of it and then move on from there. The first thing we did, we looked at a biobrick produced by Bristol in 2010, which is PYEAR attached to GFP. We ran a restriction digest with the standard iGEM, EchoR1 and PST sites. So we've got here our backbone at 2 kilobase pairs, and the insert at 1 kilobase pairs, which fits up with what we've got there. As well as running it uncut, so super cold, so it runs a bit further, and then linearized using EchoR5 coming in at 3 kilobase pairs. So we were quite happy we had what we were looking for, and we decided to move on and look at some qualitative work. So the first thing we did was we grew E. coli that had been transformed by this biobrick in media containing different levels of potassium nitrate in order to activate it with nitrates. We found nitrates to be sort of one of the easier ones to activate it with, just to get an assay of how it works. So as you can see here, it's going from 0 millimolar through to 10 millimolar. We've got quite a good gradient. And moving on from there to characterise this biobrick, we're looking to use perimeters in order to get graphs to show the exact levels of different substrates and how it affects the activity. But we felt we've got enough out of PYEAR to move on, and we decided to look at the mammalian counterpart, which is called CARG, and looked at producing a dual promoter using both.
So, as Russell was saying, to take nitric oxide signalling forwards, we decided to increase the flexibility of the system, which could sense nitrogen species. So we decided to synthesize our own dual promoter, which contains PYEAR and its mammalian counterpart CARG, which both are responsive to nitrates, nitrites, and nitric oxide. We actually synthesized 2 genes with different orientations, bacterial mammalian and mammalian bacterial, as we weren't sure how they would respond in mammalian and bacterial cells. We hope that we've got our first biobricks from these genes. We ligated together our inserts with the PSV1C3 standard plasmid backbone from the iGEM parts registry, and as you can see, we achieved colonies from these. We've now sent these off to be sequenced, but before we've sent them off, we chose a restriction site that's unique to our inserts, and we ran a restriction digest with BAMH1 to verify that we had in fact got our biobricks. And as you can see from the gel, we were able to linearise our plasmids at 2,321 base pairs, suggesting that we had in fact transformed the biobrick that we expected. And you can also see that the uncut plasmid remains circular. So we've now sent this off for sequencing, and we should have a biobrick balance week hopefully. To start to characterise our promoter, we started with a growth study, and we used an absorbance at 660 nanometres, as this corresponds to cell growth. As you can see from the graph, there's no difference between the growth of our transformed cells and the growth of our control, which is just constant cells, which suggests that our promoter doesn't affect cell growth. In the next few weeks, we hope to add a reporter to our dual promoters, such as GFP, in order to quantitatively characterise the response of our promoter to different substrates, such as potassium nitrate, sodium nitrite, and nitric oxide. Eventually, we hope to use an effector, such as nitric oxide synthase, as this has applications in cancer therapeutics. The environment surrounding the tumour is both hypoxic and it contains low levels of nitric oxide. Therefore, if we're able to target our promoter to that environment, we could synthesise high levels of nitric oxide, which are toxic and could therefore cause tumour necrosis. A system such as this would require a high level of complexity, which is why we thought of the idea of a comparator circuit.
So, as Russell said earlier, there are sensors out there for nitric oxide, but none of them are very specific. The mechanical sensors and the various promoters will all pick up other small nitrogen species such as nitrates and nitrites. Okay. We here now? Excellent. Sorry. So, as Russell said earlier, one of the problems we've had is with specificity. So, while there are promoters and mechanical sensors which will sense nitric oxide, they also sense other small nitrogen species, such as nitrates, nitrites, and some small oxygen species like hydrogen peroxide. So for this, we tried to produce something that would give a specific output based on the concentration of nitric oxide. One of the ways was to take a broad spectrum promoter, which was relatively specific to nitric oxide, which is the PYEAR promoter, and couple that with one that was more specific to, for instance, nitrates. Compare the two outputs and mathematically work out concentration of nitric oxide. Then we realised this mathematical function could be done within the cell, and so that led to the comparator circuit. So we produced two different leaders, each had a promoter and a reporter. The sections in between will complementary bind to one another when expressed. So there are two different reporters and two different promoters, but both are modular. So any different reporter or any promoter can be used. If these are put into a solution containing nitric oxide and nitrates, then the nitrates and the nitric oxide will bind to one of the promoters, but just one of the nitrates will cause transcription to the other. The two mRNAs that are then transcribed together, and you'll get because of the imbalance, because there's more nitrates, more stimulation of one of them due to the nitrates and nitric oxide, and less of the other. There will be an imbalance in the number of mRNA produced. There will be more of the RFP. These will then come together with a CFP to bind and form an RNA duplex. This will cause the ribosome binding site to be sequestered and translation to be locked down. You can see here, here's the model of the two different leaders bound to one another, and red arrow indicates the two different ribosome binding sites. The imbalance that we have, so there's 4 RFP RNAs and 2 CFP RNAs, will mean that when the two come together, only 2 of the RFP RNAs will be knocked down, and 2 of the RFP mRNAs will be left with their ribosome binding sites free for the binding of ribosomes and translation. This means that you'll get the expression of RFP within the cells. Also, this now means that the expression of RFP is dependent on the concentration of nitric oxide, leading to the expression of greater amounts of RFP and greater concentrations of nitric oxide.
So now we've kind of narrowed down our idea of nitric oxide sensing through to the comparator circuit, and we think with some characterization of the genes we should be able to find a way to accurately tell the nitric oxide levels in a system. We started looking at sort of future applications we could have of our system and the comparator circuit itself as a broader spectrum idea. So using nitric oxide sensing as well within our project, one of the main ideas we had is the cancer therapeutic. So as Joy said earlier, cancer cells tend to have like a base level of nitric oxide they try to use for angiogenesis. And we had the idea of using the comparator circuit to deliver short bursts of nitric oxide, but only for long enough for it to damage the cell rather than for the cell to re-establish the baseline at a higher level, and eventually induce apoptosis and try to kill the tumour. We also looked at the idea of cell fertilisation. So if we can not just work on nitric oxide, but work for, say, the nitrites and the nitrates, we can look at the levels of those in the soil. So for instance, nitrites rather, if they're accumulated, can be toxic to plant life. So we could use a system that can sense if they're too high compared to the nitrates, and then convert the nitrites back into nitrates to hopefully increase the fertility of the soil. We also looked at a new legislation that's being brought in by the, I think it's the BRE, the Buildings Regulation Re-establishment, I think. And they have been looking at nitric oxide being given off in construction. And they are trying to get people to monitor it because low levels of nitric oxide being given off results in high levels of carbon monoxide, and obviously carbon monoxide is pretty bad for everyone on the planet. So if we can accurately tell the levels of nitric oxide, we can know that we're not giving off too much carbon monoxide. Away from the nitric oxide, we've also seen ideas in, say, diabetes. So for instance, the circuit could look at insulin compared to blood sugar and decide itself whether we need to have more insulin produced or whether there's enough to work with blood sugar. General pollution, so you could look at the levels of, say, CO2 in the atmosphere and decide whether there's too much and then work to change it. And as a diagnostic tool, so you could put the bacteria into, say, a blood sample from a patient, and doctors can get a real-time readout of the levels in comparison to one another.
Okay, human practices. Human practices is something we've all been very enthusiastic about this year, and within the first week of our project we went and recorded a radio programme with Star Radio in Cambridge, which was very exciting. We're also hosting our own event this Sunday at one of the main venues in Norwich called the Forum. We're going to have an interactive activity day for families to come along to and explore synthetic biology. We were also in our local paper this week, and we've been producing teaching resources which we hope to use in local schools in September. So you can see there are jigsaw plasmid. So we're going to try and get across the idea to children that they can build together different characteristics to produce a final product where they'll build their own crazy creatures. We've also been lucky enough to work with an artist as part of our project called Amy Congdon, and she's helping us to produce a film which explores different ideas of synthetic biology. As you can see on all of our t-shirts, we have a symbol that's to do with our film, which we will tell you all about in Amsterdam.
So pretty much in conclusion, we've managed to get it from the broad idea of nitric oxide sensing through to this comparator circuit idea. At the moment we're characterising the dual growth around the comparator circuit, and we will have lots of data to show everyone in Amsterdam. Just a few thank yous to our advising team and support team that couldn't be here today, as well as our sponsors. Thank you for listening. Any questions?